Learning medicine is hard work, so don't learn. ImbibeMed. Before we begin, let's have a look at this. This molecule looks simpler than this, isn't it? Well, it is, and for obvious reasons. And the conversion of this bigger molecule, which is glucose, to this smaller molecule, which is pyruvate, in the cytoplasm of cells is known as glycolysis. The term glycolysis is derived from the Greek word glyco, which means sugar or glucose, and lysis, which means breakdown or dissolution. This pathway is often referred to as the Embden-Meyerhoff pathway, the EM pathway, in honor of two biochemists who made a major contribution to the knowledge of glycolysis. Glycolysis is a universal pathway in all the living cells. Let's see some of the salient features of glycolysis. Glycolysis takes place in all the cells of the body. Why is that so? Because a few organelles may be absent in a few cells and a few others may be absent from the other cells. But cytoplasm is always there and glycolysis occurs in the cytoplasm. Glycolysis occurs in the presence or even the absence of oxygen. So what is the difference? In the presence of oxygen, pyruvate enters the mitochondria and is oxidized to carbon dioxide and water. This cycle is known as the Krebs cycle. In the absence of oxygen, pyruvate is converted to lactate. Because it can function in the absence of oxygen and mitochondria, glycolysis is a major pathway for ATP synthesis in tissues that even lack mitochondria. And the tissues which lack mitochondria are erythrocytes, that is RBCs, cornea of the eye and lens of the eye. Well, is it that pyruvate or lactate is the only fate of glucose in cells? The answer is no. There are other escape routes to the glucose molecule. The intermediates of glycolysis are useful for the synthesis of amino acids and fats. Now let's dive into the actual steps of glycolysis. The pathway is divided into three distinct phases. First is the energy investment phase, in which glucose borrows energy from the cell, as is true for any new startup that takes place. Next is the splitting phase. And the third is the energy generation phase, in which it finally gives energy to the cell in the form of ATP. The first energy borrowing phase starts with the conversion of glucose to glucose 6-phosphate by borrowing an ATP. Now that glucose 6-phosphate owes an ATP to the cell, it cannot escape out of the cell. Well, this is just a small memory aid to remember that glucose 6-phosphate is impermeable to cell membrane. Glucose 6-phosphate changes its form with the help of the enzyme phosphohexose isomerase to give fructose 6-phosphate. Fructose 6-phosphate again borrows an ATP to form fructose 1,6-bisphosphate by phosphofructokinase. This is an irreversible and regulatory step in glycolysis. Now that glucose has borrowed 2 ATPs from the cell, it splits into two 3-carbon compounds, glyceraldehyde 3-phosphate and dihydroxyacetone, which are interchangeable. After this, glyceraldehyde 3-phosphate gets converted to 1,3-bisphosphoglycerate with the help of the dehydrogenase enzyme. This step is important as it leads to the formation of NADH, which later forms 3 ATP in the mitochondria. 1,3-bisphosphoglycerate, being a high-energy compound, changes forms sequentially and generates 2 more ATPs, ultimately leading to formation of pyruvate. Thus, 4 ATPs are formed from 2 molecules of 1,3-bisphosphoglycerate. Let's have an account of how much ATP is generated with the help of glycolysis. ATP or energy generation varies depending on the presence or absence of mitochondria and oxygen. For each molecule of glucose, under aerobic conditions, 6 ATPs are generated from 2 NADH molecules via electron transport chain. 4 ATPs are generated during the energy generation phase. minus 2 ATPs which were initially borrowed by the glucose molecule in the energy consumption phase. results in a net gain of 8 ATPs per molecule of glucose under aerobic conditions. The scenario is not so great under anaerobic conditions. No ATP is generated from NADH molecule. 4 ATPs generated in the energy generation phase minus 2 ATPs consumed in the initial energy consumption phase results in a net gain of only 2 ATPs under anaerobic conditions. Glycolysis is very important even in cancer cells. Cancer cells display an increased uptake of glucose and glycolysis. As the tumors grow rapidly, the blood vessels are unable to supply adequate oxygen, and thus a condition of hypoxia exists. Due to this, anaerobic glycolysis predominantly occurs to supply energy. The cancer cells then get adapted to hypoxic glycolysis through the involvement of a transcription factor, namely hypoxic inducible transcription factor. This hypoxic inducible transcription factor increases the synthesis of glycolytic enzymes and glucose transporters, thus further breaking down the glucose. However, the cancer cells cannot grow and survive without proper vascularization. Thus, one of the modalities of cancer treatment is to use drugs that can inhibit vascularization of tumors. That's all for today. ImbibeMed signing off. Stay curious. Bye-bye.